of the day, you will realize everything that we do, we are very anthropocentric in our ways, right? Whether you study plants, or whether you study environment, or whether you study microbes or animals, what are you doing it for? You want to see how it ultimately benefits the mankind, right? So that's what I mean. So everything that we do is actually with you and you alone as a centerpiece for receiving the benefits or the ultimate outcome of all this work. So that is where we talk about clinical relevance or translation. They're all kind of referred to very variedly. And unseen future, of course. You will know, hopefully at the end of the talk, why I say unseen future. Okay, quick. You have to answer. A couple of slides have kept it this way. What does this represent to you? Anybody, just answer. One look at it. Huh? Louder. Huh? Variation. Diversity of variation. So, all that I would say is in the context of health and disease. Again, very basic. We have talked about, all of you, from your school days, you learned about the DNA double helical molecule structure, etc. So what has really happened over the last 70 odd years since the double helix was described? So if you really look at it decade-wise, the 1960s was one of the earliest, nothing to do with the DNA, but they looked at, there was a possibility to study chromosomes. And when I'm saying chromosomes, plant chromosomes, animal chromosomes were done much before. Human chromosomes could be studied. You knew there was 46 in humans and not 44, 45, 48, which was like the biggest long time debate. Okay, so we had chromosomes, and Down syndrome is a classical example. You in your neighborhood or in special schools would have seen children with the Down syndrome phenotype. One chromosome extra makes it a syndrome. So you knew that chromosomal anomalies can result in a disease condition. So this was big time, and you will not be surprised to know the earliest from the 1960s, it still happens to be that one branch of science which continues to be in the clinical or the translational domain in the hospitals. You can have any amount of molecular biology and the next generation stuff which is happening, but basic chromosomal analysis remains fundamental, and it has only improved with different technologies or better ways of visualizing, but it is all there. 1970s, you know from history, recombinant DNA was when all the 70s saw all of that big boom with recombinant DNA technology because of the restriction enzymes. So that was one major shift from observational biology, which was like the first generation, and there was a little bit of observation again under the microscope or taxonomy or looking at just the anatomy. You had observation. Then came the era of experimental biology, and then we have moved on to the next experimental biology at multiple levels, whether you talk about a gene or you talk about a genome or you talk about all of the omics that we talk about in today's context. It's moved on from experimental. What did we move on to? No, I'm talking in silico. In silico or computational biology dominated the era. And from in silico biology, we have gone one level higher because you have understood some biology, you have got all your computational tools in place. So what's the next one that people talk about? Bioinformatics, part of computational. Huh? No, systems biology. Systems biology you talk about. So, every step, the technologies moved, and you also had a next and the next and the next level of understanding biology or asking questions. So all that kind of started in 70s, and then of course the Human Genome Project. Okay, so you had Human Genome Project which came, which was the culmination of all of the technologies, the recombinant DNA technologies in hand. They thought we should be able to kind of look at the human genome, and they thought that getting the sequences will be a big time and a very useful exercise. So that is what happened. Following that, you had variations which were identified. That's the differences that we said between you and me, or between whatever, I don't know. Okay, so genetic variations, but in the context of health and disease. And then you had the next generation, which was your next generation sequencing technologies. Everywhere you would have heard whole genome sequencing, whole exome sequencing, and all kinds of developments over and above that. So that was in the last 10 years, 12 years or whatever. And then little older, 1990s, the minute you had recombinant DNA technology, the minute you had some kind of disease gene or regular functional gene discoveries, people thought that they could do gene therapy. So gene therapy is a very old attempt. So 1990s already saw efforts in gene therapy and gene editing. That means you are making a change. It's exactly like you edit something. You write something and somebody edits, right? It's exactly like that. You are correcting the spelling mistake there, and that kind of has started about 7-8 years ago. So this is really been the way the things have gone over the last so many years since the double helix. And again, these are just a few introductory slides to put you in the thing. I've written there, this is a slide which was made in 2001. Okay, the next 3 slides are actually that, because when the Human Genome Project results were announced, everybody came with their own thoughts on what should be, what should not be, what will happen, etc., etc. So they said 2010, they said there will be a lot of DNA tests available for a lot of genetic diseases. Just move there. Okay, genetic diseases. And genetic variations got documented because you have lots of changes in the genome. Everybody is going to be different at multiple locations. Sequences of other genomes became available, or they thought it will be available, and they said some genes, the functions will be known for some genes. 2020 they thought will be a much bigger jump. They believed that they will have gene-based designer drugs for diabetes, hypertension, and of course cancer therapy will be big time. Personalized medicine, pre-prescription testing, that you get tested before you actually are given a medicine, whether you're going to respond, not respond, etc. All this they believed was going to be by 2020, including mental illness. Because those of you who have had an opportunity to meet kids who are mentally ill, parents are going from pillar to post to understand what really the reason for mental illness is. Okay? So everybody, is this true? Have you achieved that?
Not absolutely.
Not absolutely, no, we are not even, kind of way, maybe 10%. Okay? 2030, in a few years, they believe that there will be comprehensive genome-based healthcare, full computer model for human replacing many of the lab experiments, because you know that you can do a lot of simulations, lots of different kinds of modeling, everything is possible. Average lifespan anyway has been on the rise. They think average lifespan, which means including for an Indian who's like, in a developing country, is still going to be better. And then of course there would be major anti-technology movements, and there would be, the last one is the most important, that there would be serious concern that you will take charge of your own evolution. Okay? Now we are dependent on nature over a period. We have kind of come with different kinds of evolution. Right? I mean, evolutionary mechanisms have been operating. But this is what we said will be 2030. Some things we know, some things we don't know. We are waiting for it. So what I'm going to tell you today are brief insights into some of these aspects. I will tell you a little bit about the human genome in health and disease the way we know it today. I will tell you how to look for, if you have a family which is with a particular disease, how do you go identify a gene for it if someone has all not really identified. So that comes under discovery genomics. And while we talk about regular genomics, we have limitations. When I go into that part, I will tell you. I will introduce you to something called as IO genomics, which is particularly for complex traits. Okay, a complex trait I will introduce you in the next slide. Then of course comes the science for society, which is what we talk about translation. Whether it is just for the family or for the general public, or whether it's a public health kind of an issue, including gene therapy. And then of course the last one is a guess. So if you talk about human genetic disorders, broadly, we are not talking about infectious disorders, we are not talking about communicable disorders, we are not talking TB and COVID, we are talking about genetic diseases, that is diseases which have a genetic basis. When you say diseases have a genetic basis, you are talking about a variation or a set of variations which may be responsible for the disease phenotype. Okay, so chromosomal ones like the Down syndrome, which I already told you, is approximately 6 to 8% of all disease burden. Single gene disorders are another 6 to 8%. Complex traits are the largest. Does that make 100? No. So do you know where the other 25 is? We still don't know, right? That means there are newer diseases coming up. There are newer genetic causes for those, which one still, so clinicians are constantly keeping their eyes open to identify new phenotypes, new disease phenotypes and so on. So this is roughly about the pattern. Now, chromosomal I've already told you. When you say monogenic disorders, first years, can you answer classical example of a single gene disorder? Monogenic is single gene disorder, right? Classical example, Mendelian disorder. Sickle cell anemia, thalassemia, cystic fibrosis, Duchenne muscular dystrophy. Name it, it's one gene, one mutation which runs and is responsible for the disease. Okay? Unlike that, complex traits are multiple genes contributing together with a large dose of the environment or the non-genetic factors. Okay? So complex traits are, your classic examples are diabetes, hypertension, cardiovascular diseases, asthma, rheumatoid arthritis, and a whole lot of them. So first, Down syndrome I already told you. You know very well that all that you have is one extra chromosome 21, so easy for you to identify. So chromosomal disorders are very easy, very possible to kind of identify all of the chromosomal anomalies in a fetus, and therefore if identified, the fetus can be medically terminated. Okay, medical termination pregnancy is permissible if you know there is a chromosomal defect. This is how simple it is when you talk about single genes and uniform risk for all those who have it. On the other hand, the complex disorders, we don't even know. If you ask me a question, how many genes for type 2 diabetes, I have no answer. Nobody will have an answer for you. Okay? We don't even know, despite all the advances, how many genes are there which are responsible. And therefore, testing for a gene or susceptibility is out of question. But just to explain, I have just put 4 A, B, C, D, and I've also kind of done this here to show the impact of variation. It's not just going to be the change, but it's going to be determined by the environment. Like, just to give you an example, if you're talking about a family full of type 2 diabetes individuals, if you are a part of the family and if you're going to take extra care of your nutrition and your exercise and so on, you may have all of the bad genes, but you may not develop type 2 diabetes. That's where the environmental factors, and we talk about gene-environment interactions which are responsible for the complex traits. More importantly, if I have said 4 genes, it's not that the same 4 genes are going to contribute to the disease in every disease individual, affected individual. If this is A, B, C, D in this order, the A, B, C, D contribution could be this in this order. That's what the color codes say. So it is lots of genes of minor effect and cumulative contribution of those which are responsible for the disease. So you already see how difficult complex traits are, and therefore you will understand why, despite all advances, we are not made any progress with that. So with that introduction and clarity on single gene disorders versus complex disorders, we then move on to why do I need to know all of this? What is the central dogma in molecular biology? DNA, RNA, protein with one reverse arrow, right? This is the central dogma in medical biology, right? You have prediction, prevention, cure and treatment in that order. Okay? When I say prediction, you can't predict just looking at an individual. You have to have a biomarker, you have to have a genetic test, whatever. So if I'm talking about genetic disorders, I will refer to as a genetic diagnostic test. Okay? So if you have a marker, you can do a prediction. If you can predict, you can prevent. If you can't prevent, you know the use. Prevention is better than cure. But if you can't prevent, you hope for a cure. Have you known any disease condition, particularly complex traits, where, or even single gene traits, where there is a perfect cure? Anybody? Most of the times, cure is very wishful thinking. We end up only with treatment or management. Okay, so that's where we are even as of today. There are some curative ones, but they are really far and few. Okay, so you have this also referred to in today's context as P4 medicine. So P4 stands for predictive, preventive, personalized and participative. Okay, so you all these terms are quite interchangeably used, but that's really what you're trying to do. So the first exercise then becomes discovery, because if you don't identify a gene, you're not going to be identifying a mutation there. If you don't identify the mutation, there's no way you can talk about prevention. Okay, so as far as I'll put the questions first, so you will know at the end of the talk whether we have achieved, or you can already answer. Do we know all the genes in the genome? Do we know all the disease causing genes? Do we have genetic testing for all diseases? Why do people respond differently to drugs? You go to, you have a headache, I take one tablet maybe of aspirin, and somebody says, no, that just doesn't work, I need 2 tablets of something else. Somebody will develop vomiting, somebody will develop some other symptoms. So these are all individual differences. Do you have biomarkers for all the diseases? Will obtaining protein structure solve the problems? And so on. What are the broad answers? No is an answer. Uniform answer across is no. And now I'll ask you, if I have to get the answer yes for any of these, then you can always say, why bother about humans? I can look at animal studies. Or your argument could be, anyway, the West is far more advanced, so why can't we just copy from them? Why should we invest our money to do our science here, or reinvent the wheel in a country like India? That could be your argument. So I'll give you 2 counter arguments to that. This is only for fun that I put, that if you look at man and mouse, okay, that's the chromosomes of humans, the spectral karyotyping image that you would give, and this is what is done along with the DNA from the human. If you hybridized it with mouse chromosomes, this is the kind of a pattern that you get. What do you see? Lots of rearrangements have been there before it evolved into humans and this. Anybody telling me what is the most noticeable in this picture? Look at all the chromosomes and quickly tell. The X and the Y. Perfect, lovely. Okay, so the X and the Y have been so highly conserved. Okay, while a lot of other things have gone. And this is true for all mammals. Many genes are conserved on the X chromosome. And many of the genes which are expressed in the brain, 40% of genes which are expressed in the brain are all in the X chromosome because of its conserved nature. It also is maintained that way because in the males you have an X and a Y. There is no recombination at packaging. With the result, your linkage group just goes happily and it just stays. It's only in the females that you have recombination. Okay, so there are many advantages. So this is what it is in terms of the relationship between man and mouse. Having said this, if there is so much of difference in the linkage groups, okay, there are 40 chromosomes versus 46, that's not important. If they are in different linkage groups, the regulatory elements and everything else which is going to be responsible for expression is going to be different. Okay, this is again a fun picture just to tell you. This is for the question, why can't we use animals for answering things? Why do we require humans all the time? Okay, this is what it is. You have a lot of similarities, but you still can't have things that you're studying in humans because human phenotype is indeed different. Okay. Now, why can't you use populations? Why can't you use something which is, Caucasians? Americans have got just about everything, or Europe has everything. Why can't we just do that? If you look at a Negroid race or a Mongoloid race, you can tell them. You will say, okay, this person is Mongoloid race and so on. But science is all evidence-based. You require something else. So when you are looking at populations, you use genetic markers, okay? And there are a set of markers which helps you identify the different populations. So extreme left, so these are all the codes that you give. Japanese in Tokyo, Yorubans in Ibadan, Caucasians from Utah, Chinese from Beijing, Gujaratis in Houston, and North India is NI. These are codes that you give for different populations. Now if you use that, set of markers and you try and do the population mapping, you see this is Africans. Far away from there are your Caucasians, the blue. Chinese and Japanese, perfect overlap because they are a Mongoloid base. And this is Indians. Okay? The green is North Indians. The orange inside is Gujaratis in Houston, which is West India, you can kind of just say. But perfect match. And if you see, they are more closely related to the Caucasians rather than the two other population groups. If I just talked about North Indians and South Indians, again you can have the difference. Now the picture is the other way around, but you just see here, North India is closer to Caucasians and South Indians are just adjacent below. So normally you hear the word, those of you interested in population studies and so on, you always hear people saying North Indians are more Caucasoid in origin because your features are more like, the Caucasians and so on. So you can actually see the North Indians maps closer to the Caucasians compared to the South Indians. And you can go on and on. Just to tell you that whatever is there in different populations, while some things like single gene disorders may be perfectly there, all translatable across ethnic groups, everything for complex traits, you'll have to be very cautious because I said gene environment interaction is what is important. Your geography is different, your food, your diet is different, everything is different between populations. So while some basic genetics may be common, you might have to restrict yourself or focus on specific populations to get the answer specifically for complex traits. So that's very important. So you can actually look at patterns for that. And the last one for this part is, why is there a disparity between different population groups? All of you know there's much higher consanguinity in Indians. Naturally, the chance for small a coming together is going to be much higher because you're having consanguinity, known or unknown. And then, of course, there's selection pressure, socio-cultural practices, etc. And here, you have very different kinds of environment and risk factors which are responsible. And so what can India contribute then? Instead of saying that we pick up anything from the West, what can India contribute? Nowhere in the world. Of course, earlier Italians used to be there, Spanish with their large families with many affected individuals. But not anymore. India still happens to be at the top of that list with large families with many people affected, which means it's precious material for genetic analysis, and large families with diseases. And then of course you have lots of different challenges which are there, which is your geographical diversity, epidemiological data is not there, overall lack of awareness, okay, and poor follow-up, etc. So currently many things can be done. You can look at transethnic settings, etc. And today one big time thing is to look at the gut microbiome, okay, because you are what you eat, okay, and most of the diseases you believe are very easily controlled by controlling your diet. And that's just kind of on the side that I want to just tell you. Now I'll come back to the big jump, the first big jump. So you had recombinant DNA technology, people were ready to take it to the next step, and they said Human Genome Project. They said we will sequence all of the genome very quickly and go through double helical molecule. You want to sequence all of it. This is a typical karyotype. So you want to sequence 24 chromosomes, and they said the short-term aims were 3.2 billion base pairs would be sequenced, and you will have all of them annotated, and you will have gene functions assigned to a large bit. That was the short-term goal. They claimed as soon as the Human Genome Project was announced in 2000. Okay. Long-term was gene therapy, pre-prescription testing, and of course basic research, which will always continue. So there was a lot of joy because they thought the minute the draft sequence is announced, they thought all old age diseases will be conquered, and everybody was happy that you will have no diseases because you have understood the genome and you will get everything. But unfortunately, this is where we are. I hope the lights are not too much. You can see the slides clearly? No? You can't see. So I think you should turn off some of those backlights. You know that I should have told you that before. Oh, you should have kind of switched up. Okay. Anyway, never mind. These are just unexciting GATC. Is this better or also? Yeah, just keep one or two lights in the center on. Otherwise, just turn off the others. Okay. So you have very unexciting, not all of them out. Yeah, just keep last one on. Last one on, please. Okay. Yeah, that's perfect. Hopefully it's a bit better on the other slide, but this is just to say that it's a very unexciting GATC in whatever combination running into pages. Now a small exercise for you. Take a language textbook, take off all the punctuations, etc. Try and see what it reads like, and can you make a sense out of it? Quick, quick.
One day
I heard
last.
one day last. Then you're complete. I heard one day last April from everybody. Okay. After that, start backwards. Try and see. Alright. Okay. So this is what it is. This is exactly where we are with the human genome, even 20 years down the line. We are still trying to make sense of what is there in the genome. Large parts of it we have understood. Like one day last April, everybody knows these words, everybody knows these letters. No matter where it is, you can just say it. But if you don't know something, to identify a word from the letters and to make sense of that word is very, very important. And that's exactly what we're trying to do even as of today for 98% of the genome. 2% protein coding, we know it all. But we don't know the regulatory elements even for that 2% of the coding genes if there are changes. Okay? So you have so the major fallout was not in identifying all of the disease genes, but the major fallout was identifying the spelling mistakes in the genome. They looked at 5 different individuals, but at each one they found different sets of spelling mistakes. And that's when the labs actually were fighting with each other. You must have done something wrong. But later they realized these were single nucleotide polymorphisms, or called in short SNPs, which are present in all of us, except monozygotic twins, who will have identical pattern. Everybody else will have a change. You will have 50% of the change from your father, 50% from the mother. Your sibling will have a different 50%. So unless you are twins, everybody is going to have a mix and match of these changes. So the SNPs became a great handle, and you will understand it that all SNPs may not be functional, all SNPs may be just little cosmetic change. If I said a car and a cab, is there a difference?
Not much.
No, you mean the same thing, but there is a R and a B, okay? If I said I'll take a bar, completely different word, and normally I'll take a bar unless you meet somebody you don't really say that phrase in you. Or I'll take a jar, completely different meaning. Now if you look at what happens in amino acids, TCA is serine. At the wobble, you have the TCC, which is serine. But if the T gets changed to ACA, it's threonine, CCA, proline. So you can imagine you have an opportunity for one marker or one spelling mistake to change into any of the other 3, which may change the amino acid and therefore the protein, or it may not change. So you call it as a synonymous change and a non-synonymous change, and this is what is being extensively used now as markers for us to identify different genes, because these become like your landmarks to reach the gene of interest. So you can have insertions and alternate spellings. Color, COLOR, the British and the American style of spelling, or a S and a Z and so on. So these are just easy ways to remember what it does, and you have long stretches of DNA sequences with many, many, many changes which are then contributing to haplotypes. But if the change is one which is causing a disease, it's like just glutamic acid to valine, one base change, but it's resulting in a sickle cell, and that's a disease. Okay? So what we know now is there are lots of different SNPs, of course, are plenty. There are blocks of segments which are repeated multiple times, which are copy number variations. You have insertion deletions, and now we talk about rare variants again, which are supposed to be unique. So there is a big talk about rare diseases and rare variants and so on, but only this little red portion is your protein coding gene. Everything else is absolutely non-coding or intergenic, intronic, etc., etc. So you have more than 10, actually now it's even a 20 million SNPs. Every day there are newer. More human genomes you sequence, more SNPs you're finding, okay? But that kind of opened up, instead of answering the question for the human genome and the genes, it actually opened up a whole lot of omics era activities. Earlier you said genetics or a gene, now you say genome. That means a larger picture. You're looking at the entire genetic constitution, okay? So you have functional, pharmaco, computational and so on. Together with that, you also have, all this could be just using DNA. Here you have tissue specific. Transcriptome is going to be tissue specific, metabolome or proteome, anything. There are ever so many newer omics which are coming up in the now. So this is not important. I do a lot of different things, but my primary aim is of course to look at discovery genomics and wherever possible to try and do translation. We look at complex traits, we look at single disease traits, we look at the biology, we look at, just about, that's broadly what my lab does. Now, why should you be doing discovery? So just to tell you, this, look at this is a 2019, it doesn't matter, even today if you Google OMIM, that is on Mendelian inheritance in man. If you look at that site, it will tell you how many genes are already known and how many of them you have the Mendelian phenotype. You say there is this disease running in the family, but you don't know the gene, okay? So the gene hunt is on, molecular basis unknown. In 2016, it was 1611 which were unknown, and now you have 1557. That means every year there are more and more disease causal genes which are getting identified, okay? So that's true for all of them. Just to tell you that we are still a long way off before giving a gene or ascribing a gene to a function with a particular disease phenotype. So discovery genomics continues to be at the very central area of medical genomics. So I won't go into the methodologies, but just to tell you what can happen in families. Here is a family. You can obviously see first generation, next generation, and so on. By the age itself, you can make out. The family is full. It is an X-linked disease. Therefore, only boys are affected. Okay? And you can see how large the family is. That means all mothers are carriers, and they ended up with a child boy who's affected because single X, and therefore the disease one. So these are routine things which come to us for diagnosis, etc. So if you don't have a diagnosis with a known gene test, you need to go and identify the gene, and we were lucky. We did lots of different things, but ultimately we were able to identify a gene called MID2. And we were like, probably this was the very first gene, disease causal gene, which got discovered from India using the next generation sequencing technology. Okay? And this is a part of OMIM. Every time you discover a new disease causal gene, it gets updated. So MID2 will be there as a part. Now for X-linked mental retardation test, MID2 is a part of the many genes which are on the panel. So that's the way you go about and use a family. You will never get a family like this elsewhere, because the minute you see a disease is running in the family, they'll say, look, we rather adopt a baby rather than have our own baby. But that's not the culture nor the philosophy in India. So you'll say, oh, one more try, and that one more try is shown here as an example. 4 boys affected, 1 girl unaffected. Okay, that's only sister unaffected, 4 boys. Okay, the family just produced children, and you just kind of had many children. So there again, identifying the gene here was very important, not for these kids, because they are already intellectually disabled or challenged. Okay, it was very important for us to find that for this girl. If she had the same mutation on her X chromosome, if she were going to have a baby boy, there's a very good chance that she will get it. So you had to kind of find. This is the utility of doing these kind of tests. Okay, so we again identified, but this took us the longest time to find. It took us almost like 7 years to find the mutation, because it was not in the exon. the mutation was in the splice site. You know, you talk about exon-intron junction, it can contribute quantitatively. The protein amount will be different, so you ended up having an intron-exon splice junction. But normally when you're doing, you're just looking at an exonic mutation, you know, coding variation, and you put everything else in the bin. But when you don't find answers, you go back and revisit, and that's what we did. Now, I don't know how I'm doing with time. If you
This 323.
323. Okay. No, because I have a little film here for showing you Parkinson's disease patients. Okay, young onset. Here is a family where consanguinity is there.
Yeah.
Parents are related, so you end up with consanguinity 2, 3, 5, 8 children, 3 of them affected, and look at the age of onset. All of them in their teens got affected. So, do you want me to?
Yeah, please.
Young onset Parkinson's disease. Okay.
हाँ जीप दिखाओ हाथ ऊपर करो दोनों हाथ ऊपर करो चल के दिखाओ चलो छोड़ दो इसे ठीक से चलो ठीक से चलो चलो
So you got approved, right? Much more blank, right? The boy at least was responsive.
हाथ को उठाओ हाथ को उठाओ हाथ उठाइए दोनों उठाओ उठाओ ये हाथ उठाओ सीधे हाथ को उठाइए
She can't really do. Okay.
पैर सीधा करो पैर सीधी कर लो
So this is what you say, you know, when you have Parkinson's disease, you So this is a kind of a family. They had 8 children, 3 affected, and consanguinity. So 2 bad copies of the gene came. And obviously the family is just running from pillar to post to find out what really is responsible for the disease. So this is again something which came from AIMS. We were able to identify. So we have had about half a dozen different new disease causal genes that we identified using families which came to us through the clinical collaborators. And there is so much more scope for you to identify new genes. It's just that the doctors have to be aware that here is a family which is not responding to, which is not giving you any clue from a known test, but it's there. These are details of how you go and do next generation. All these are from next generation sequencing. Conventionally, if you're doing, it would take you maybe 5 to 6 years to get it. But luckily, these were done in about 10 months. You already had. And once you have a gene, you will establish the basic function. Otherwise, if you don't show the basic function, nobody will publish. They'll say, oh, by chance you have that variant. They will not believe it's a disease-causing one. So in today's context, no genetics is just looking at family, looking at genes, trying to look for a mutation. If you have to understand function, which is all your functional genomics, it is a very, very integrated approach. Whether it is structural biology, animal work, computation, cell biology, biochemistry, you need all of this. And therefore, those of you felt bad, you didn't get into honors, you don't have to worry. As life science students, you have so much opportunity to learn all different aspects, and the best of all from the 3 departments. And the way your scope is so much more. Absolutely. So, you really need to kind of appreciate that you're going to have a much nicer way to look at it. Okay, unlike that, the complex traits are a little different.
Excuse me, ma'am, can I interfere? Yes. I was wondering, like, while you were talking about this Parkinson's, I know of a family where a person got Parkinson's at 50. So were genes dominant by the time, and suddenly, he had an accident, and then Parkinson's and moved on.
So
what do we attribute it to?
So normally Parkinson's disease is an old age of onset. When you get familial cases, you are almost sure it is a young onset, because that's when you have 2 bad copies of the gene. Otherwise, Parkinson's falls into this category of a complex disorder. So if somebody got it at 50 or 60 years of age, that's a normal. If there is physical insult or chemical insult, your chances of getting Parkinson's is much higher, because your neuronal cell gets damage to the neuronal cells is much higher, so you will manifest the disease symptoms. So there are multiple factors. That's why we say multifactorial. Not just genes, but environment also, which contributes, which gets triggered. And that environment could be anything. It could be from stress to lots of pesticides that we have, etc., which can all result in neuronal health or neuronal disease. And you have the polygenic trait. I said multiple genes are contributing to that, okay? So we know that there are many problems, but the way you study is using a chip. Instead of looking one snip at a time, you have a chip. There are 100 samples that you want to study, use 100 chips, do the hybridization, look for the pattern, and then do a simple test of statistics. A simple chi-square test is what you do. You're just looking in case group and control group, just to tell you that the risk allele should be higher in the case group. That's what you see, okay? So if you see that you have a marker like that, then you say, okay, the disease has got some kind of a connection, or this marker has a kind of a connection with the disease. So that's broadly where we are with complex traits. But we now know 20 years of all of this work, we haven't got any answers, as I told you right in the beginning. We have lots and lots of genes which have been identified with lots and lots of diseases. Each of the colors represent a different kind of a disease group: digestive system disorder, inflammatory disorder, neurological disorder, etc. It gives you a big picture that one gene is not associated with one disease, but you're talking about shared underlying biology across different diseases. So if I say this particular marker, this is associated with 8 different diseases. So it is underlying disease biology which we were getting to understand, that there are shared pathways and shared biology, which if you go back and think, inflammatory disorders, you definitely will have to have a shared biology. It doesn't matter whether you're talking about inflammatory bowel disorder, or you talk about rheumatoid arthritis, or you talk about any other inflammatory condition. You know that some pathways are going to be the same. So this is where we are, and that's when we decided that we need to have alternates, and we think one of the alternate is to use the Ayurveda system of classifying people into 3 broad groups of Vata, Pitta and the Kapha. So that's like the basic philosophy is different. When you go to an allopath, what happens? He's treating your symptoms, okay? And all the exercises, you treat the symptoms and you get cured. In Ayurveda, the philosophy is maintenance of health and prevention of disease. And most important is, of course, I told you that you have 3 kinds of people. This is all Google stuff, you can see. Vata, Pitta, just to tell you that this is the way it is. If you went to the clinic with 10 people, this is what everybody is, put into one basket in an allopathic clinic. In reality, all of us here will fall into broadly 7 groups, but 3 are the main ones which are shown here. Others are mixes, okay? So if I talk about the 3 main groups, I already have a beautiful way of subgrouping or getting homogeneous ones. For me to get my genes of interest, I have a greater chance that if I did the subgrouping, I can get. So this is something which we have been following in the few years, and this is what I've been pursuing very, at this moment, because we think this is what we should be able to do to get better answers, irrespective of the technological advances and so on. Ultimately, you want biomarkers for disease and you want newer therapeutics. That's what you're looking for at the end of the day. So anything that we do, we think that a multidisciplinary approach, but with a starting point, which is kind of to get people into different groups. For which again, if you have biomarkers, then there are no arguments. Otherwise they'll say, oh, that doctor called me Vata Prakriti, somebody is saying I'm Pitta Prakriti. There will always be this debate and distrust, okay? Okay. So, you have to have very, very integrated approach in P4 medicine. You have to look at all of the omics for you to get the big picture or to get the answer for a complex trait. So only then you're going to be able to do. Now I'll go quickly. I've said these things before. Genetic diagnosis is then the next part. How many of these findings can actually be translated? I already told you that there are multiple things you can do. Chromosomal testing, mutation testing, etc. IVF, in vitro fertilization is big time. when you're doing in vitro fertilization and identifying embryos, they can also see what are the diseases which are there in the embryo. If a family has gone because they had a disease, they have resorted to IVF, they definitely want to get screened for that disease, mutation, and so on and so forth. Okay, so I'll just tell you one example with newborn screening for inborn errors of metabolism, which is like a larger relevance. This is straightforward, where chromosomes, I told you, you can have fancy methods to study chromosomes where you can look for translocations, because you can see perfect reciprocal translocation, color change. These are all visually very easy for you to identify, and this is in routine clinical practice. You can do prenatal diagnosis and get biochemical tests, you can do genetic tests, you can do chromosomal tests and identify the thing. But if you talk about a larger context, that comes to specific families. But if there are children born with inborn errors of metabolism, that means you can actually treat the child who is born with the disease. You don't have to know anything about the gene. You don't have to spend time looking for the gene, because it's only based on a metabolite profiling. And there are 600 genetic diseases which fall under the broad category of inborn errors of metabolism. I'm sure in your basic genetics you would have all learned inborn errors of metabolism, which are generally autosomal recessive disorders. You don't need the genetic information, but you just have to have a heel prick of the baby 24 hours after the baby is born or after the baby had its first feed, because you're looking at the baby's metabolic profile, looking for the metabolite differences. You can already identify if there are any variations, do a confirmatory test, and if there is intervention or therapy possible, you can immediately do that. And yet we don't do it in our country. 50 years, it's 50, 60 years now that we have inborn errors of metabolism, but we do very little. It is not even mandatory. Only if your neonatologist or a pediatrician is smart enough, you say, hey, look, something doesn't look right with the child, let me quickly get a test done. But many kids will just go unnoticed, or they will all get mentally retarded or get any of the phenotypes. So we had an opportunity to look at 200,000 newborns from across 20 hospitals in Delhi state with the clinical collaborators, and of course the results were absolutely fascinating. We saved approximately 1 in 200 newborns, and it's a classical example of translational medicine without any cost, okay? Without any cost and without any kind of a fancy methodology or a technology. It's a simple LCMS based run that you can do. And indeed, it is a technology for the common man. Okay, these are the kind of conditions which are all treatable. Okay, and yet you don't get to treat them at all because the doctor completely ignored and did not get the basic test done. Not the doctor's fault. It is generally the system's fault. It's everything. These have to be. This is a classic. I don't even have a SIP pair from India whose picture I can put. This is obviously, you can see the mongoloid child, right? This is the first child born to the family. Okay, who was congenital hypothyroidism, lack of thyroxine, got mentally retarded, dwarfism. These are all the characteristics. The minute that happened to the first child, the family woke up, the doctors woke up. The second baby, again born with the same mutations, but luckily put on treatment, just thyroxine treatment right from the first week of birth before the damage, because the treatment has to begin very early on. So phenylketonuria, you would have all learnt in your class book, PKU. All that you need to take off is phenylalanine off your diet. But to get phenylalanine free diet in India, a poor man or a lower middle class can't afford. With the result, you just live with a mentally challenged child. And you might have more in the family coming because you didn't know what it was. So this is what actually happens. These are a very sad example of congenital adrenal hyperplasia. One gene, one mutation, 7 rupees tablet a day is all that these people require. These are born with ambiguous genitalia. If the doctor is aware and immediately gets them to test, they can put them on treatment. They don't do it. So this group of people are very familiar to you, right? Part of them whom you see are the ones which are CAH positive. Okay, but unfortunately they did not get treated. Otherwise they can be just regular as a part of, you will not even tell if anybody is sitting amongst us, because they would already be on treatment. But because it's a steroid drug that they take, they will be a little immunocompromised, but that's about it. Okay, so this is really the sad part, where we don't even have a policy which brings us to getting this kind of stuff. So this I can skip. Okay, so you can actually do that for just about, $1,000. In the lab it costs only 1,000. So even if a commercial company is doing it, maybe they'll charge 3,000, but still it's worth it. They're testing you for about 50 different conditions. Okay, which is like such a doable thing. Okay, I'm kind of coming to the end just to tell you that pharmacogenetics, I refer to pharmacogenetics time and again, right? Pre-prescription testing. Like your eye card, you could have a little card in your pocket which will tell the doctor which are the medicines that you're going to respond to, what you're going to not respond to, whether you're going to develop adverse drug reaction or not develop a drug reaction, etc. Okay, so this is what you expect, and this is all pharmacogenetics and pharmacogenomics, a very, very doable kind of a thing which can happen. Okay, now gene to medicine, these are just examples that I'm telling you, because I told you that I will give you end to end, just to get you an idea. So there was a classical example of a standard epidemiological survey, where one African American woman was found to have very, very low levels of the bad cholesterol. So it was just an epidemiological survey which was done, but they identified that at the lowest level, the next thing that they did was they went and looked at the genetic makeup, and they identified a mutation in her which was responsible for that really low level of bad cholesterol, which opened up the market in a big way. So that gene is gone. Go read for yourself. PCSK9. Okay, classical example of one gene, one mutation picked up from an epidemiological study, which kind of got them into business. So now it is several $1 billion business within 2 years of getting the gene. Everything is always not so hunky-dory fig-paste. It's like a lot of effort that you've heard from me. But there are things like these. So serendipity is a part of all science and the excitement in all science. Okay, so you can have this kind of a thing. So just that one, they made inhibitors out of that. So if you give that and you use that for people, you can control your cholesterol, and it's big time market now. Okay. Now finally to translational efforts.
Ma'am, is that commercially available?
Yeah, at the moment they are already marketing.
Okay.
They're all marketing it. Okay, but you need to get tested for what your genetic makeup is, etc. And there again, although it's one particular gene resulting, you could have interacting partners which can make small differences. So one needs to look at it holistically. So I've told you about chromosomes, I've told you about DNA testing for mutations, I told you about inborn errors of metabolism already in terms of translation. But here I'm just telling you that you could also have gene therapy, where you're talking about putting the right form of the gene and hoping that the person is, the child or the adult is going to get cured. I won't go into details, but you can do it in vivo, or you can also do it ex vivo. Then final one is somatic editing. Today you're talking about editing, right? So these are just to tell you, cystic fibrosis is a classical example where first generation gene therapy was tried, second generation is tried, now the third generation gene therapy is happening with genome edits. Okay, but that genome edit is restricted to the individual who is going to be affected. It's not something which is going to be passed on from the individual to the next generation, because you're only talking about somatic cell gene editing for correction. Okay, so these are details just to say that you can have ex vivo, in vivo therapy, etc. But biggest challenge has been to get the right vector and the right method to put the vector inside the body, because invariably vector targeting or delivery is the most difficult part. This is what the editing was all about. You all know that Charpentier and Jennifer Doudna got the Nobel Prize, and all that they used was the beautiful thing that they identified in the bacteria, which got translated to humans, and that's how you can actually do all of the cutting. I won't go into the details, but you can understand. And so, if today, okay, quickly among the students, tell me what this is. Okay, the teacher said already. Okay. So that's exactly where we are. If we talk about a somatic cell therapy, no issues, because it happens in an individual. Your cells are taken. Suppose it's a bone marrow. Suppose it's a hemophilia. You will just take thalassemia. You will take the bone marrow. You will edit. You will go ahead and do whatever you want to do. The individual is being treated. When he is dead, whatever has happened, even if there are off-target effect of the CRISPR-Cas9 technology for gene editing, nothing is going to happen, okay? But if by chance you're talking about germline genome editing, which is what our famous Chinese scientist did. He took in vitro fertilization embryos, he edited them for a gene called CCR5, which is a gene susceptibility gene for HIV, and promised the family that he will get them babies who are never going to get HIV and AIDS. So he went and manipulated the embryo. But the technology of CRISPR-Cas, we are not yet there that you have no off-target effects. While it has the on-target desired effect, we don't know how many off-target effects. Of course, you can argue and say, do a whole genome sequencing, you can identify. But when we don't know what the whole genome sequences, the 98% of them actually code for or what they do, how do you know what these off-target effects are going to be? Okay? And therefore, there is no way you can do germline genome editing as of today. Okay? It is absolutely not allowed. He did it. He went behind bars because the whole world was, against. Because obviously he did not respect the ethical committee clearances. We don't know what actually happened, but all that we know is that he presented it in a meeting and everybody was shocked, and that kind of really brought about this huge furor over editing, germline genome editing. So the futures for anybody to guess. So if that is, so you remember one of the early slides I said, 30 years down the line we don't know, we'll take charge of our own evolution. Because people want to have beautiful babies, people want to have intelligent babies, people want to have a fair baby, dark baby, tall baby, short baby, and so on. So if this is what you're wanting to do, you don't know what you're ending up with, okay? So that's where, while genetics is absolutely fascinating, a lot of things are possible, a large dose of caution is always going with that, okay? So that's your designer babies. We know that you could have eugenics, you could have all kinds of stuff which is going to happen. And there was this commission which I happen to be a member of, and they brought out very strict guidelines on no to human genome germline editing. And if at all parents are so desperate to have a biological child, then they have identified categories of people who may resort to editing, but with a large dose of doubt. So my conclusion will be incomplete if I don't tell you about the ethical, legal and social issues in medical genetics. So if you have a bad gene, say for example breast cancer gene, you have a mutation, you're already you have your genome makeup, you go for a job or you go for an insurance, they say, oh look, you have a BRCA1 mutation and therefore I will not give you a job because you are a liability to my company. Or the insurance will say, I'll charge a higher premium because you are already going to be a liability. That's just one example of that. Fetal sexing, big time. In India, it's the largest culprit. Everybody wants a baby boy, okay? And they'll go any end to get this going, okay? And that's again something which is a punishable act, but these are all issues which you have to be very aware of. While you have the technologies in your kitty, you need to be also aware of these things. Human rights, of course, big time, on what do you do, what you should not do, how much right. A mother, for example, who knows she has a Down syndrome baby, she has a right to decide whether she will bring up the baby, but that means go through with the pregnancy and bring up the baby, or it's entirely her decision whether she wants to go for a medical termination of pregnancy, okay? So these are all issues of human rights. And then of course all that debate about how you can take your own evolution into your hands. And then of course all this, I have lots and lots of collaborators and lots of students who kind of do all the discovery work and all the things that we do in the lab. And this is my message for myself, okay? Because there is so much to do, while there is really a lot. And the message for you? That's a message for you. Sorry, tell me the time now. 4 o'clock. 4 o'clock. Sorry,
4:50.
We enjoyed it thoroughly, every bit of it.
Ma'am,
We
I
enjoyed it.
have
Thank you so much. Yeah.
While you were talking about this genomics,
please turn on the
so
lights.
how do you respond to today's Lady Harding demonstration? Today there was a demonstration.
Everybody please maintain silence.
Ma'am, today I read the newspaper saying that Lady Harding had a big time demonstration against all this. So how do you confront and respond to these problems?
Sorry, I missed you.
I'll read
Okay,
exactly
I
what I Lady Harding case.
Yeah, what did they say?
किसी का खुल रहा है क्या?
What
It was today's newspapers this that there was a big time demonstration against these kind of practices. Considering Ayurveda as one domain
this,
area,
okay.
and they've started up a clinician.
No, I'll tell you. This debate is always going to be there, because I just told you, unless we have biomarkers, which will really help to put the people into 3 major groups, you will always have this debate. It's the best P4 medicine which has been there. Ayurveda has been the best P4 medicine which has been there, 5,000 years old. But unfortunately, no scientific validation. You can all hear me at the back? Okay. So there's no scientific validation, okay? So the whole effort today is that we need to have scientific validation of this, wisdom that they've had over the years, where it was purely based on observational science. But everything that they said, they knew exactly what diseases you are likely to develop. They will give you food recommendations. They will also tell you what your outcome will be on a treatment. We don't have, and we today talk about precision medicine. We are, so many thousands of years down the line, we don't have any of the answers. But the unfortunate thing is, if you do not have scientific validation, if you don't put the method of scientific validation in place, then what Lady Harding doctors are doing is absolutely right. All that you want to do is get a Ayurveda clinic in every place. Yes, I also went to the Ayurveda clinic at Holy Family Hospital. Those of you know Holy Family Hospital, it's one of the top allopathic hospitals. 25 years ago, they already had a department of Ayurveda, because they realized that you needed to have a holistic approach for health and well-being. Okay? Not just treatment of symptoms, but you needed to have maintenance of health and well-being. People who don't get cured of rheumatoid arthritis, they're completely crippled. They go to the Ayurveda clinic because they have a stomach cleansing called Panchakarma. Today, if you look at the science, you know what the science is? What does Panchakarma do? Changing the gut microbiome. Okay, it gets all your system rid of the microbiome, and it gets you, depending on each individual, they will give you particular treatment which repopulates your gut. And that is why people go back to the Ayurveda clinic where all allopathy has failed. So, to answer the concerns, yes, you can't thrust something on another group which has been practicing just because you want to make it popular. If you want, you should actually have many schools who are doing big time Ayurveda research, giving validation, identifying biomarkers, which I will give it to you in writing, which will be far more easier if you have the subgrouped people rather than looking for a biomarker in tens of thousands of diabetes patients. It's not going to work. But if you have clean subgroups, it will work. So this is where we need to do things with caution. It says IMA protest. IMA, you know, right? Indian Medical Association. protects against mixopathy. Know all about doctor strike against garments moving, allowing surgeries by Ayurveda students. Okay, now really speaking, this is also ignorance because there are only, sorry, I want to take this one thing because she brought up this thing. There are only two documentations. There are only two documentations in Ayurveda. One is called Charaka Samhita, another one is called Sushruta Samhita. One is for the physician, one is for the surgeon. If 5,000 years ago they had beautiful, like Leonardo da Vinci pictures, you see, every vein, everything which is seen, they knew exactly what operations to be done. They only did it with practice and observations. Same thing with a physician. Just by feeling the pulse, they already know what you're likely to develop, what your current situation is, etc. But if you don't have validation, so those documents were all there, they systematically got destroyed thanks to invaders. But now it's a word of mouth, and there are very good Ayurveda schools. But I think if these things have to be put into practice, so if an Ayurveda student is doing surgery, there is nothing wrong. Because if he has understood the Sushruta Samhita correct, he will be doing better surgery, I would dare to say, than somebody who's trained regularly. Because he's understood the nuances of it from his own text. So that should be the, that's the unfortunate thing that we don't have validation. So anything to promote and validate will be the way to go. So the question was, what is the process involved in discovery genomics? Number one, there has to be a family, because when we talk about identification of a gene, picking up a sporadic one case is going to be difficult, although in rare disorders that's what they are trying to do. But given the limitation of poor knowledge about the whole genome, you would rather have a family which is having a disease, where you will have at least a couple of members affected with the disease and a couple of members not affected with the disease, and of course the parents. If you have a family like that and the clinician says, look, this looks questionable, they'll put DX question mark some syndrome, and you do all of your molecular tests, you don't get an answer, because when you send the DNA test for a DNA test, they will look at a panel which is already available in the testing lab. If a gene is not in the testing lab, they're going to say, sorry, none of them match with what is in the panel. So that becomes your first material for discovery. The second thing is, you see what is the mode of inheritance. Is it an autosomal dominant mode, autosomal recessive mode, etc. If it is a dominant mode, then you will have a different method of analysis of the results. If it is recessive, it's a different method. Okay? Now, how do I reach that place to get the method in place? Today I will only do whole exome sequencing. Because if it is a phenotype, which is apparent disease, you know very well that I can look at affected individuals, unaffected individuals, and compare the DNA sequences of these. When I look at the sequences, what is my first assumption? This is all in the kitty which goes, okay? The first assumption is that if it is an autosomal recessive disease, I should have two small a's. That means it should be a homozygous mutation in the affected. Parent should be heterozygous. And unaffected ideally should be just wild type. Okay? So if it is an autosomal dominant, my mode will be, I am only looking for one mutation, capital A, which is shared between one parent and another child. Now you say parent was not affected, but we don't know. That's where you look at the pedigree and decide. And if you then look at what is the pattern of that mutation, go and look. Everything is available in the database now. For the genes, which are 20,000 odd genes, are already known. They are already shown in which tissues they express, broadly what function it has, all that's already known. So now the first question is, let's assume it was a neurological disorder. What's the target tissue you expect to be involved? Brain. Right? So first thing you will do is go look at whether the gene is expressed in the brain or not. If the gene is not expressed in the brain, you will put it on a back burner. Right? And then you will move on. So most important. So one is having the appropriate clinical material. Second, when I say clinical material, I say informative clinical material. Two, very good quality of sequencing data. That not that parts of the sequences are missing, because then you will miss the clue. Okay. The most important will be the knowledge of the inheritance pattern and the assumptions you're going to make for picking up the gene. Like that is where a biologist doing bioinformatics and a computer scientist doing bioinformatics for medical diagnostics, there's a world of difference. Because you are applying your biology, they are just applying their technology. Okay, this is the difference. So you need to kind of think. So once you have done your bioinformatic analysis, you will find some genes. You may not just be lucky to find just one, you will find a set. Then you will have a narrowing down, like I told you, target specific pattern, which tissue is it expressed and at what stage in development is it expressed, etc., etc. And then of course you have to do your functional genomics. Functional genomics are in vitro, in a cell line. Then you will go for animal model. Then you will look at, if possible, anything which is an ex vivo kind of a stuff. But normally, at least the in vitro and an animal model would be the way to discover a nice new disease causal gene. That's the pathway to go.
Thank you so much, ma'am, for sharing your insights. And now I would like to invite our co-convener, Dr. Pratibha Pant, to extend vote of thanks.
Thank you, Sahil. Good evening, everyone. On behalf of the Life Science Society, I take this opportunity to propose a vote of thanks to all those who are present here. First and foremost, I would like to thank the speaker of today's session, Professor V.K. Thelma ma'am, for accepting our invitation and delivering this lecture here today. Ma'am, your presentation was so wonderful. You were so energetic throughout the talk that we really enjoyed it. Thank you for sharing your knowledge on human and population genetics, your work on various genes which cause human diseases is really very impressive, and I'm sure it will inspire many young minds present here. I thank you once again, ma'am, for your support and coordination, because of which this event is a successful one.
I didn't even thank you for inviting me. I didn't deal in any of these formalities and I just went.
I would also like to thank our principal, ma'am, Professor Rama, for being the catalyst that stimulate us to do our best always. I thank Professor Vijayarani Rajpal, teacher in charge, Department of Botany, for her constant encouragement and support and standing as a pillar of strength for all of us. I would like to thank Dr. Riddhi Khurana, convener of Life Science Society, for planning and organizing this talk. I would also like to thank all the faculty members who are here with us for sparing their valuable time for being here for this event. And an event like this cannot happen overnight. It requires a lot of planning and hard work. I would like to thank Kasturi and Shubham and their entire team for tirelessly working for this inaugural lecture, because without them it was not possible to organize this inaugural lecture. Finally, I would like to thank all the participants. I would like to welcome Dr. Sudeshna Majumdar, University.
Please sit here. Sit now.
Ma'am, that's okay. I would like to thank all the participants for their enthusiastic participation in this program and making it a success. On that note, I wish happy learning. Thank you all for your presence. Thank you.